Bom, it's a pleasure. Natacha Gillet. Natacha Gillet. Uh, she's from CNS and also was a student at the Universidade João I. Natacha foi, então, estudante da Universidade João I, trabalha com química molecular, mecânica molecular, simulações em reações orgânicas e tem agora no laboratório de química NS de Lyon, enfim, com uma produção muito grande, principalmente agora com os últimos trabalhos em photodamage and repair in DNA, em biologia computacional. Então, eu gostaria de passar a palavra à professora Natacha. Muito obrigado. Thanks. Yeah. Please, Natacha. Natasha teve uma queda na conexão, mas ela já está entrando novamente na sala. Só peço que aguardem mais alguns minutos. Já.
Does that work? Yes, okay, thanks. Sorry for the delay. I was with Firefox and it didn't work. Okay, thanks a lot for the invitation. It's a great pleasure, and it will have been a bigger pleasure to come in Brazil. Now it's cold and night in France. So yeah. I will talk about our studies in our group about damaged DNA from double strand to the nucleosome damage using molecular dynamic simulation, only classical for today. So first of all, why we are interested in DNA damage and at which scale? So we are chemists, so of course we are looking for a molecule, and we work with bDNA, so the double-strand DNA, the classical one, which everybody knows, where you have damage formation between nucleobase or involving one or two nucleobases. And when you study this double-strand DNA, you can have this sequence effect or the effect of the flexibility of the double-strand DNA. But in our cells, DNA is not really like that. It's wrapped around a histone core to form the nucleosome, the primary unit of the chromatin. And then in nucleosome, you have mechanical constraint in different electrostatical environment. And now what we want to know here is the damage hotspot in this system involving both DNA and protein. And then what other people do at higher level is to see the impact of damaged DNA, deformation of damaged DNA on the chromatin, or and then you can study with data from cancers the mutations in nature on the whole chromosome, and see the periodicity of the formation of damage, or in which sequence you can see some damages. So which kind of damage we are interested in? You can have the photo damages, such as which are created by UV irradiation. So you can have CPD, so cyclobutane pyrimidine dimer involving or2 thymine or thymine cytosine, and you have bond between C66 and C5C5. Or you can also have the 6,4 photo products, where you have a bond between two thymines and one is oxidized into pyrimidone, and you have bond between C6 and C4. Other damage we can be interested in are damage due to oxidation of DNA. They will appear mostly on guanine, because guanine has the lowest ionization potential among all nucleobases. And you can create radical guanine by first oxidation of guanine, radical cations that will lose a nitrogen. And then this radical guanine can react with water for molecule to form atosoguanine, or with protein or other molecule to form crosslinks, such as DNA crosslinks. Other damage can be abasic site resulting from a partial repair of DNA, which can also be linked to proteins. So the chemistry of this damage is highly combinatorial chemistry, because you have the issue of the sequence effect. The sequence can have impact on the flexibility, but also on the transfer, for example, on exciton transfer involving cytosine and thymine, or an electron transfer involving guanine or adenine, mostly guanine and also adenine. Then you have the structure impact. For example, if you look at this G quadruplex, you have several guanines which are stacked, and if you create a cation there, its lifetime will be much more longer than in B strand DNA, as it can be transferred between the different guanines, so it can stay about 1 millisecond, whereas it's 1 micro in beta strand DNA. You can also have the mechanical constraint due to the nucleosome, for example, or the interaction between the different strands in nucleosomes, not only along the sequence, but also between these two wrap part of the DNA. And you have also the problem of the environment about solvent accessibility, which can be more or less the same, but we have minor groove in beta DNA. But for nucleosome, for example, you have one part which is close to histone core, other part which is solvated. You have tails that can interact with DNA, and also you can have a protein which interact with DNA and impose some specific exostatic environment and also some constraints which can help to the formation of CPD damage, such as this transcription factor, which constrains the position of the two nucleobase here, and that increase the rate of formation of CPD damage. So the methods we use to do that is mostly classical MDs at microsecond timescale with hundreds of thousand atoms. We'll use the classical force field parameters for bonding and stretching, for dihedral, for the intermolecular interactions, for Coulomb energy, Lennard-Jones, and for this, we use Amber force field and sometimes dedicated force field for our damages. If we want to go further in the reactivity, we use QMMM. That means that we select a few parts of DNA and we treat it with DFT or semi-empirical approaches methods to have the behavior of the electron. And this quantum parts should be quite small because quantum simulation are a bit expensive. And then we treat the environment as classical, so for water, the rest of the biomolecules and the ions in the system. So our objective is to understand the formation of the DNA damages, then to characterize the behavior of damaged DNA, to understand how the repair protein can interact with it, and the impact of the sequence, and so on, the environment on it. And finally, the repair mechanism for recognition, interaction with DNA, and also mechanism. The final goal is to have a picture of DNA lesion of spot. Just take a look, is there no issue? Okay, so for today, I will speak mostly about 6,4 photo products. So this is damage, as you can see here. The two thymine are not in the coplanar. The pi stack is lost. It's mostly perpendicular. That's why this lesion is considered as inducing a high flexibility of the DNA damage, of the damaged DNA, sorry. So it is due to UV irradiation. It is known to increase DNA flexibility and facilitate high bending of DNA. And you have different proteins that can repair it. And in humans, it's repaired by the nuclear excision repair process involving different proteins, such as, for example, this RAD4, where you can recognize the damage and make the adenine facing the damage extrahelical, and then the damage will be also extrahelical and then repaired by another protein. We wanted to have a look at the impact of the sequence, the closed sequence, only the flanking nucleobase close to damage, on the flexibility of this area, but taking into account the whole oligonucleotide. So we have chosen 6 sequences. So the one used by the experimentalists for this this structure. So it's AAG, the inverted one, so TAC, and other sequence that were used in previous studies, so TAT and the inverted one AAA, and CAT and GAA. So the difference are that the fact you have a stack of purine, which can be a better stack than the stack of pyrimidines or pyrimidine purines. You can have also the impact of to have GC, or which has 3 hydrogen bond between nucleobase, or AT, which has only 2, so it's less strong, or also having the sense of the polarity of your sequence. It's because you have the damage is not symmetrical, so you have a difference if you put a G here or a G here. So then, for the methods, we can use only classical molecular dynamics, but even if we go to microsecond, it's quite difficult to be sure that we sample correctly the conformation, and in general we can be blocked in one conformation and only visit it. So one solution is to use Replica Exchange, where we perform several replicas at different temperatures, with different temperatures, and they are running at the same time. So you use as much as GPU as you have a replica, and then periodically you make some attempts to see if you can exchange the conformation between 2 replica using energy parameters. So our rates were between 29% and 40%, which is quite good. And to decrease the number of replica we required, we used so REST2, where you only hit the DNA and not the whole box. That means that water stay at 300 Kelvin, and you can change the parameter of your DNA molecules to reach an effective temperature of 500 Kelvin. So then, we have 6 molecular dynamics of 1 microsecond. We look only on the trajectory at 300 Kelvin, and we make cluster analysis to get the different clusters, the different representative conformation of we have in our trajectory. So, for example, you have here for AAA and TAT. So you can see that the first cluster is quite similar. It's just the pi stacking in the undamaged strand is conserved. The Watson-Crick hydrogen bond 2, you only have 1 more hydrogen bond between the thymine 34 and the adenine 16. And for TAT, you have an additional hydrogen bond between the alcohol of T34 and this hydrogen atom of thymine 15. The second cluster also gives a similar conformation, where the adenine with a nucleobase in position 18 is rotated and come into pi stack with the pyrimidine moieties of the damage. And then you have also the adenine 17, which rotates. And you have pi stack between 15, 16, and 33. And a nitrogen bond is created because 33 and 34 are coplanar, so you have nitrogen bond here. For AAG and TAC, for AAG, you have the same main cluster as before, and for TAC also, but for AAG we found only one cluster. So the first idea was that it is less flexible than the other one, but then it is clear that it appears that this cluster is wider than for the sequence. That means you have in this cluster you have more conformation which are close but more flexible in one part. And for the other cluster of TAC, we have similar from TAT, only the difference here is that adenine 17 stay in pi stack with 16 and 33. And for GAA and CAT, so for CAT, we have the same clusters as for TAT and TAC. So that shows that if pyrimidines flanks the adenines, the sequence does not affect so much the flexibility and the conformational space of our damaged DNA. And for GAG, we have the two first similar damages, and then we have only a last cluster which is different and highly distorted with this strand which is close to 90 degrees. This high band can be found for all sequences, and even in normal AMD, it's really highly flexible. You see that you can go from 0 to more than 90 degrees. And for AAG, it's a bit more higher than for the other one, but we have really, really large distribution, highly flexible DNA, which is really useful for recognition protein binding. So the question was, why is this difference with AAG? So there's 2 points. Of course, first, you have a CG pair here, where you can have these 3 hydrogen bonds, which are highly conserved during all the dynamics, even using Res2. And what you don't have is the hydrogen bond we observe in TAT, and which is present in TAC and CAT and GAA. So this bond this hydrogen bond increase the rigidity around the 36 and 15 nucleobase. And as you can see here, when you don't have this hydrogen bond, so for AAA and AAG, you can increase the distance between these two nucleobases and lose the hydrogen bond with the same. So you can have several conformation on this side of the damage. That could explain the different behavior from AAG, which has rigidity on this part of the damage in 5' and more flexibility on this side in 3', this side of the damage. So we look for extrahelical conformation in our MDs, and then we test them. You're running 100 nanoseconds for each extrahelical geometry we found, and we have some stable, that are present for more than 50 nanoseconds in our MDs for AAG, TAC, and GAA. But for AA and TAT and CAT, we have no stable extrahelical conformation. This one is very interesting because if you draw together this conformation and the one from the X-ray structure with Radford, so the one that I showed before, you see that you have some similarities, saying that we can find conformations that are quite close to experiment and with proteins. So it's very encouraging for the following. So there is really a large flexibility around 6-4 damage, and you have an impact in sequence impact, at least for this flanking base pair. And the fact that we use Res2 allows us to find different geometry, not only with normal AD, we have only the first cluster. And the X-ray helical conformation can be compared to X-ray structure and led us to think that we can explore the flip out mechanism. So then, I would like to talk a bit about some interactions that are possible with a repair protein, which is photolyase. Photolyase belongs to the big family of cryptochrome photolyase. Cryptochrome are involved in plant photomorphogenesis, in regulation of circadian rhythm, and perception of Earth's magnetic field. I was more interested in PHRB, which is 6,4 photolyase, and which belong to a part of this family which is close to the ancestor of all these proteins. So unfortunately, we don't have photolyase as mammals, but because they have really efficient to repair or CPD if they are CPD photolyase, or 6,4 photoproducts if they are 6,4 photolyase. So the damage is flipped in the active site, and then you have a flavin cofactor that can be reduced, fully reduced, and when the reduced FAD is excited, it can transfer an electron to the damage, and then you have for 6,4 proton transfer from active site histidine, bond rearrangement, and then you have the two thymines, and the damage is repaired. The damage here for bond rearrangement is still under debate, and the protonation state of the histidine is also questioned. In PHRB, it's quite interesting because, first, you have several residues on the surface that can stabilize the interaction between DNA. So again, the DNA is bent, and the protein. So you have arginine, lysine, and the highly polar proket, and this histidine that can be inserted in the space led by the flip out of the damage inside the active site. So yes, PHRB is quite interesting because it's sensitive to the concentration of divalent cation in the middle. Divalent cation are really essential for DNA repair, but this is not the case for other 6,4 photolyase. It's really has an impact on repair and not binding, and they don't have the X-ray structure of PHRB with DNA, so they don't know where can be the cations, but they know that if they mutate these two aspartates, the repair rate is really decreased. So we tried molecular dynamic simulation of the system, and what we observe is that two magnesium cation can interact with this aspartate molecule when we have a positively charged histidine. When it's central, we have only one cation inside. And if we mutate, we observe impact on the presence of the cations. For example, if we mutate this aspartate, there is really no more cations here. So the cations are close to the damage, and they can interact with the damaged lesion and facilitates the electron transfer by putting a positive charge close to the damage, which will be negatively charged after the electron transfer. So if you go to have a look on the electrostatic interaction, you see that really it's electrostatic interaction between the magnesium and the diamine is higher when you have positively charged histidine, and it's with the aspartate is lost when you mutate it to the aspartate to asparagine. And if we compare with the structure of other photolyase, what we see is that the cations are close to the position of two positively charged amino acids. So in arginine here, or lysine, which in the structure is interacting with this part, but in AMD, they observe that it can turn and come here and interacting with this phosphate. So they really have the same position. So cations are playing the same role as this positively charged residue, and the difference is really seems to be related to the evolution in this protein family. So for the three conclusions we have from this is that the evolution in 6,4 photolayers family involve or divalent cation or positively charged residue in the same position, but this positive charge can help to the electron transfer can help the electron transfer to the damage, and the positively charged histidine is a better candidate in PHRB for the repair of 6,4-pp. And last, I would like to tell you about nucleosome and the 6,4 photoproduct in nucleosome. So to be quick, in the nucleosome, you have a DNA segment of 146, 147 base pair, which are wrapped in nucleosome around an histone core. You have 8 histones, so two dimers of H2A and H2B, and 2 dimers of H3 and H4. And this histone has a very flexible N-terminal tails with positively charged residue, so lysine and arginine. And these tails can have an impact on the properties of nucleosome. So these are the crystallographic structures, so the tails are extended, but of course in solutions that can be they can change their core formation and interact closely with nucleosome. So for 6,4-pp, we use the crystallographic structure of the damaged DNA. The 6,4-pp are or close to the histone core, so it's the in position, or placed here close to the solvent, and so the out position here, in out. We use the experimental crystallographic structure where the tails are cut just because they are too flexible to be seen in the crystallographic structure. We have 4 microseconds for our simulations, and we can compare them with the free DNA with exactly the same sequence. So we tried first to compare the bending and some entropy of the 2 strands in the different condition. So if you have the free DNA, so you see that the bend is as before, as we observed for REST2 simulation, really large distribution. While for nucleosome, because of the mechanical constraint due to the histone core, you are here really a narrow distribution, even narrower than the controlled free DNA, and which is not centered because the DNA is naturally bent in the nucleosome, so it can be flat like free DNA. And for entropy, which is quite interesting, that our inner strand, so the one which has a damage for in, or the one which is undamaged for out, can be less flexible compared to the free DNA, which is which is what we expected. But for the outer strand, the one which is solvated, well, close to solvent, it's more flexible around damage than in free DNA in these different cases. So it suggests that you have a higher flexibility close to the damage, and this can be recognized by the repair protein once again. Here you can observe that you have 2 out 2. It's because when we perform cluster analysis, we observe 2 different conformations that we will explain in the next slides. And that's why I put these 2 different states. So from cluster analysis, sorry, it's not good here, but we observe only one cluster for the two inner position and for the one of the output position. So the common thing is the fixation of the DNA at this point by 3 arginine from H2A. So arginine 32, arginine 17, and arginine 20. And then you have other interaction that depends on the length of the tails, because the tails are not completed. For example, you have the N-terminal on threonine 16, and here is on alanine 14, and here on lysine 15. And you can see that when you have the lysine 15 on the N-terminal here, you can interact with the outer part of the DNA with the phosphate, and again with the lysine 15 here for the first position of OUT1. But if you go to OUT2, the tail now goes to arginine 11, and arginine 11 can also interact with the phosphate, but also with the damage in 2 different positions. And these 2 different positions correspond to 2 states revealed by cluster analysis. So here you keep the good pairing of DNA, and you see that the lysine is here and can interact with the pyrimidine and this adenine. Then is rotated a little bit and interact only with this pyrimidine, and then this corresponds to another conformation where the pairing is lost. You have an orphan nucleobase here, and these 2 thymine are interacting with these 2 adenine. So it's true that its own tail can have an impact on the flexibility of DNA, and that we can go further with this histone tails and try to simulate the full tails, even we don't have not so much information about their conformation in solution. So that's the goal. We have still a large flexibility around 6,4 for the product, even in nucleosome, and particularly in the outer strand, even if it has or not the damage. Indeed, the UV-GBB protein, which is able to recognize the damage on the nucleosome, it can twist the DNA even if the damage is in position to have it outside. So this flexibility on the outer strand can help the protein to recognize where we have the damage. But now we need to put the full tails in the system to really know which are the interaction. So these are our perspectives. So the large sampling of DNA, and particularly the histone tail, and even because they are really sensitive to a post-translational modification, and this modification can be involved in the interaction with protein, in the structure of chromatin, and so on. So we also want to study this post-translational modification. And my main goal now is to study the DNA oxidation in nucleosomal DNA. So the modification of the guanine, the creation of the radical cation guanine, its evolution, if there are some charge propagation. And for that, I will use a QMMM approach and machine learning to analyze all our data, because we will have a lot of data from classical MGs and QMMM simulation. So this work was possible thanks to several people in my lab at the Laboratoire de Chimie de l'ENS de Lyon, with collaboration with Antonio Monari Lorraine, with my previous supervisor in Karlsruhe, Markus Esner, our collaboration in Valencia, in Prague, and thanks to the computational center in Sines. And I would like to thank you so much for your attention. So yes, if you have question, I don't speak Portuguese. Sorry. Okay.
Well, let me. It's not really a question from one of the I think this. It's well, she said temperature can break hydrogen bonds also. Probably about the first part of your talk. I think it's only a suggestion that temperature also can break the hydrogen bonds.
Yes. What we observe is for the rest of the DNA, it's quite stable. The fact is, when you use the replica exchange, if you look only the trajectory at 300 Kelvin, you will have only conformations that are you can have at 300 Kelvin. It means that you can go up, and if the DNA find another well in the surface, it can go there, and then you get it in your trajectory at 300 Kelvin. But if you look at the 500 Kelvin, of course, you will have really strange conformation. But the idea is to overcome the barrier. If you look at the trajectories, it's not continuous. It's really changing from one conformation to another one. It's just moving when you have with your atom. It's
okay. We have another question here. What is the temperature difference between consecutive replicas in your replica exchange?
So I don't remember exactly because I used different lambdas or different effective. So it's a bit different. It's increased with temperature. So yes, it's so we have 32 replicas, and it's between yes 300 Kelvin and 500 Kelvins. Yes, three.
It's good, this online version is good that we could also use the notebook to see the questions.
Yeah.
Well, so you think it's 5 the difference?
Yeah.
Between, okay. Let me see another one here. I have read in some articles that people with vitiligo, I don't know what's vitiligo, have some types of defense against UV radiation, which albino people do not. Did you understand?
Yeah. Yes, it's true that there are some proteins that can protect from UV radiation. I'm not a specialist of that, sorry. I read sometime, yes, but I was more interested in DNA damage. And then
okay, I understand.
Yes.
Let me see. There is another two. You have the chat also.
Yes.
Okay. Well, next to one or so only suggestions about this DNA relation. I think. Let me see here. There is another question here. Probably there's an invitation. I put here another question from Adam Petina. Erika. If I got it right in your last part of the talk about the microsome, you have observed the different flexibility of the image or not the image strand when faced the stone core.
Yes. Could serve as an indication for, yes, we can suppose that. We have no proof for the moment, and it will be better to have the Eastern tails to really to really be sure that it's more flexible or not. That so something that we're very cautious with this conclusion.
Yes, I understand.
But our first result, and also the crystallographic structure, because the outer strands was not well defined in the crystallographic structure, the idea was that it's probably more flexible than undamaged DNA, and the protein can recognize this flexibility or bulky lesion.
Okay.
I would prefer to have the full tails to be sure.
Yeah, I understand. It's a conclusion that I have the same question, but you have to be careful.
Yeah.
Well, I think we have finished the questions here. There's no more from the audience, and thanks again, Natasha. It was really exciting scan of DNA damage. It's important. It's really important. And well, thank you.
Thank you again for the invitation, and thanks Erika a lot.
Okay.
Okay.
We'll prepare to the next talk.